Hello all, hope you're doing well. In this video, we'll discuss about an important pathway that all cells partake in, which is the catabolism of fatty acids. Now, catabolism is a word that means breakdown. So, it's one half of metabolism. The other half of metabolism is anabolism, which is the joining or synthesis of a new molecule. So this video is going to focus on how fatty acids are broken down in a cell, and one of the major pathways of this breakdown, which is beta oxidation in a fatty acid, which is again a major component of cells, which is palmitic acid. So we'll be focusing more about the beta oxidation of palmitic acid, but initially we'll also talk about what are the other oxidation pathways or the other catabolic pathways that are seen in a cell. There are four major types of biomolecules that are found in a cell. Those are the carbohydrates, the proteins, lipids, and the nucleic acids. Now, lipids are formed by the esterification of a fatty acid, which is actually a long chain carboxylic acid, with alcohol. So, when we have the fatty acids combining with alcohol, what we get, that product is what we call as a lipid. Now, the chain length of the fatty acid can differ, so can the chain length of alcohol, and based on their combination, we have a variety of lipids that are present in nature. But then, one of the most common types that we find is a triglyceride. Now, triglyceride is formed by the combination of alcohol, which is known as glycerol. So, glycerol is a 3-carbon alcohol. As you can see over here in the picture, it is a 3-carbon alcohol. And to each of the carbons of the glycerol molecule, we have a fatty acid that has been attached. Now, the fatty acid side chain could be the same or it could be different. So what you see as R over here could be the same molecule that is there, or the R could be different. If the R is different, then we call it as a mixed triglyceride and so on. But then triglycerides are a very common type of lipid that you find in nature, and they are in fact one of the major energy storage molecule as well. Now what we're going to see in this video is how the cell breaks down these triglycerides. So when I say they are breaking down the triglycerides, it means the cell has to generate the glycerol and it has to give out the free fatty acid. Only if it gives out the free fatty acid can the fatty acid be broken down. So we are going to see what happens to the fatty acid when it is being oxidized in the cell. But before we get into what happens to the fatty acid, we need to know a bit about what are the different types of fatty acids that we find in a cell. Based on the presence of double bonds in the fatty acid chain, in the R side chain, we have two types. Those are the saturated fatty acids and the unsaturated fatty acids. Saturated fatty acids are those where the carbon has been completely filled with or saturated with hydrogen. That's why it is called as saturated fatty acid. There is absolutely no space for a double bond. So saturated fatty acids are usually solids at room temperature, and one easy way for you to remember is the triple S formula. Triple S because saturated fatty acids are having only single bonds and they are solids at room temperature. On the other hand, we have unsaturated fatty acids. Unsaturated fatty acids are not saturated with hydrogen. That means somewhere in the chain there will be double bond in the fatty acid. Now, there could be a single double bond that would be called as a monounsaturated fatty acid, or there could be multiple double bonds which are called as polyunsaturated fatty acids, or PUFA, that is the PUFA, which is again a different topic for us to get into. You can watch this video for knowing more about the different types of fatty acids. So the oxidation of fatty acids, or you can say the breakdown of fatty acids, can occur in different regions of the cell. And please let me remind you, these are all the regions that are found in a eukaryotic cell. In a prokaryotic cell, because they don't have organelles, all of it happens in the cytoplasm. Now in a eukaryotic cell, the major site for oxidation or the major place for the breakdown or catabolism of fatty acids is the mitochondria. That is where all the enzymes are present. The other important place where, or the other important organelle where oxidation can occur is the peroxisome. We will see how there are different types of the oxidation. So peroxisome shows two types of oxidation mechanisms, and then we also have a minor pathway that is occurring for the medium chain fatty acids. That is usually the fatty acids which have 10 to 12 carbon atoms in them, and that is in the smooth endoplasmic reticulum. So there are different types of oxidation mechanisms and each one of them is occurring at a different place in the cell. The three types of oxidation that are seen for fatty acids are the alpha, beta and omega oxidation pathways. Now these three pathways are mainly based on where the cut is occurring during the breakdown or during the oxidation or the catabolism. If you see over here, this functional group, the C double bond O's, and here this is a palmitoyl CoA, that is the CoA group has been attached. But if the CoA group was not attached here, we would be having the OH group. So that particular functional group is how we identify a fatty acid. It is basically a carboxylic acid at the end of the day. Now, next to the functional group, if the cut occurs, that is, if the cut is occurring, if the oxidation is occurring at the alpha carbon here, right over here, then that is what we call as the alpha oxidation. And alpha oxidation is seen in the peroxisomes. It is usually seen for the carboxylic, the fatty acids which we take in through our diet. For example, phytanic acid. So those acids, those fatty acids are broken down by the alpha oxidation pathway in the peroxisomes. And here only one carbon is lost. That is, this functional group carbon is being lost in the form of carbon dioxide. The major oxidation pathway in the cell is the beta oxidation. Beta oxidation occurs at the beta carbon. So that is over here between the alpha and beta carbon. This is where the cut occurs. And as you can see, two carbon atoms are being lost over here. So when you have alpha oxidation, you have only one carbon dioxide coming out. Whereas when we have beta oxidation, we have a two carbon compound that is getting released. That is in the form of Acyl-CoA. So this is seen in the mitochondria. This is also seen in the peroxisomes. And the beta carbon is what undergoes the oxidation. The carbonyl group, this particular group, the C double bond O group, is what is getting released along with one more carbon atom, like I said, in the form of Acyl-CoA. The third type of oxidation, which I said is a minor pathway for oxidation, that is omega oxidation pathway. This is mainly seen for the fatty acids which are medium chain, that is those which have around 10 to 12 carbon atoms, and this is seen only in the smooth endoplasmic reticulum. So if a person is having a defective beta oxidation pathway, then their cell will go for an omega oxidation pathway. In omega oxidation, the carbon is being removed from the most distant part of the carbonyl group. So if this is the carbonyl group, the cut would occur over here at the far end. That is what we call as the omega oxidation. Now, like I told you at the beginning of the video, this video we are going to be focusing on the different steps of beta oxidation pathway, especially for palmitic acid. So let's get into it. There are three steps that are involved in the beta oxidation pathway. The first one being mobilization from the adipose tissues. Now, adipose tissues are the region of our body where the fat or the lipids are being stored, and the adipose tissue are made up of cells called as adipocytes. So in the adipocytes, we have fats being stored in the form of triglyceride. Like I told you earlier, we have triglyceride which is stored over there, and from there it has to be moved to a particular target cell which needs to break down the fat, which requires it for energy maybe. Now the target cell could be any cell that has a mitochondria except the RBCs which lack mitochondria, and the brain cells where due to the blood-brain, initially we used to think it is because of the blood-brain barrier that the lipids or the fatty acids are unable to pass through, but now we know that even in the brain cells we don't see any beta oxidation because of various theories. One of the theories is that there is more oxygen that is used in oxidation, and that might make the neurons more hypoxic, or the reactive oxygen species generated are very high, which can cause a lot of oxidative stress to the neuronal cells, or even for the fact that the structure of the brain itself is made up of lipids. So it's a risk to have beta oxidation over there. Whatever it is, the mobilization of the fatty acids from the adipocytes to the target cell can occur to any target cell except the RBCs and the brain cells. Other than that, it can go anywhere for breakdown. The second step is activation of the fatty acid. We need to activate it, we need to charge it, you can say, in a way, and then it has to be transported into the cell. So here what we are talking about is transport into the cell and finally into the mitochondria of the cell, because that is where it is going to occur. The last step is the actual oxidation part, which occurs inside the mitochondrial matrix. So the fatty acid has to move from a different tissue, that is the adipose tissue, into the cell from into the cell, it has to get inside the mitochondria where it is going to be broken down. So we'll do each one of these steps in detail. The first step is the mobilization from the tissue, from the adipose tissue into the target cell. Like I told you, target cell could be any cell which needs to break down the fatty acid. The only target cell that are not possible, the only cells which cannot act as a target cell are the RBCs, because they lack a mitochondria, and the brain cells. So the fatty acid is now going to get transported from the adipose tissue till the target cell. We'll see how. Now the first step of beta oxidation is the transport of the free fatty acids from its place of storage, which is in the adipose tissue, to the target cells. Now the target cell could be a liver cell or a skeletal muscle cell or a heart cell, or actually speaking any cell which can carry out beta oxidation, which has the enzymes for beta oxidation. But the problem is that in the adipose tissue, the fatty acids are not found freely, because if they were found freely, they would always be escaping from the cell and moving all over our body. So in the adipose tissue, these fatty acids are stored in the form of triglycerides. Now we have already seen what triglycerides mean. Triglycerides are molecules where there's a glycerol moiety that has been bound with 3 fatty acids. Now these triglycerides have to be broken down to release the glycerol from the fatty acids. Glycerol is what keeps the fatty acid within the cell, doesn't allow it to escape from the cell. So we need to first break down the triglyceride into glycerol and the fatty acid. This is carried out by an enzyme called as lipase. Now lipase enzyme is found in the cytosol of the adipose tissue cell. Adipose tissue cell is called as an adipocyte. So in the adipocyte, say this is the nucleus of the adipocyte, these are the fat globules which are present, and in the cytoplasm we have this enzyme called as lipase. Whenever there is some kind of a trigger or a signal by the body, lipase gets activated. So what are these signals? These signals could be hormones such as glucagon or epinephrine, or it could just be some dietary compound which is activating the lipase. Now when lipase gets activated, it immediately breaks down the triglyceride into glycerol and fatty acid. The glycerol goes for other pathways such as gluconeogenesis, or for triglyceride synthesis, the glycerol is taken care over there. The fatty acid is now free to be released into the bloodstream. So all this what I have shown you here is happening in the adipose tissue over here, and from here we have the fatty acid traveling through the bloodstream and reaching the target cells. Now when it reaches the cells which are going to metabolize it, or which are going to show the beta oxidation, the problem first is in entering the cell, because as we know, the cell is surrounded by a plasma membrane. Now the plasma membrane is having lipids in it, so it is actually very easy for lipids like fatty acids to enter. But the problem is many of the long chain, especially the long chain fatty acids, have a negative charge on them. Because of their negative charge, they cannot diffuse easily through the plasma membrane. So they need to be transported across the plasma membrane of the target cell by using some kind of transmembrane transporter proteins. So we have transporter proteins which are involved in transporting or in bringing in the fatty acids which are found in the bloodstream into the target cell. This is the first step, wherein they have now moved from the adipose tissue which was found in the maybe under the skin or around the internal organs or between the muscles, wherever, whichever adipose tissue is now being mobilized. From there the fatty acids have now entered the target cell. But then this is just the first step, because in the target cell they again have to cross the next barrier, which is the mitochondrial membrane, and then only they can be broken down. Now that the fatty acid has reached the target cell, it has to get activated and then it has to pass inside the mitochondria. So let's see how that happens, because we need to first activate it. It is still in the form of a nascent fatty acid. We need to make sure it is up and ready for getting broken down. So for the activation of fatty acid, what we do is we combine the fatty acid with a thioester compound. So basically we are introducing a thioester linkage over there. How does that work out? It means this would be the fatty acid. This is the R side chain. This is the C double bond O functional group. And to that, we are now going to have a thioester linkage. This is what the final product of activation looks like. So this, what you see over here, is what now we call as the fatty acyl-CoA. The fatty acyl-CoA is basically the fatty acid which has now been combined with a CoA group. That is a thioester linkage. Now to do that, we have the enzyme fatty acyl-CoA synthetase, or you can just call it as Acyl-CoA synthetase. This is the enzyme which catalyzes the conversion of the fatty acid, as you can see over here, this fatty acid, in a two-step reaction to form the fatty acyl-CoA. So what happens over here is the fatty acid combines with, you can even write it in the form of a single step. What happens here is fatty acid combines with the CoA molecule, the coenzyme A, and gives us the fatty acyl-CoA. But during this process, so when this conversion is taking place, or when this thioester linkage is being formed, we need energy. Now that energy comes from ATP. So we have one molecule of ATP being broken down, please be very careful about this, into AMP. It's not one high energy compound that is being broken down, but there are two linkages being, two high energy bonds are being broken down in the same molecule. So we have ATP getting converted into AMP, and we have the pyrophosphate that is being released. So this is a very important thing, because we need to keep this step in mind when we are calculating the net yield. Now this entire reaction, so in this step we have the CoA getting in, we have the ATP getting converted into AMP, that is adenosine monophosphate, and the inorganic pyrophosphate, and we finally get our product, that is fatty acyl-CoA, which is now the activated form. And like I said, this is being catalyzed by the enzyme Acyl-CoA synthetase. Now this step can occur either in the endoplasmic reticulum, or it can happen, usually it happens on the outer membrane of mitochondria. So at the outer mitochondrial membrane, that is where normally this process takes place, but other than that, it can even happen in the endoplasmic reticulum. Now once this is done, once we have the activated fatty acyl-CoA, now we need to see how it can get inside the mitochondria. So we have only reached till the mitochondria. It needs to now get inside for the final oxidation to occur. Now that the fatty acyl-CoA has been formed, this has to now get in through the membranes of mitochondria and reach the mitochondrial matrix for beta oxidation. So this, what you're seeing over here, is actually a blow up of this particular region. We can see this is the outer membrane of the mitochondria, and on one side of it we have the cytosol. Then we have the inner membrane of mitochondria, and the outer and inner membranes are being separated by the intermembrane space. Inner to the inner membrane, we have the mitochondrial matrix where all the enzymes are present. So the fatty acid, which is now in the activated form of fatty acyl-CoA, has to traverse or move through these two membranes and reach the inner part of mitochondria. There are channels in the outer membrane of mitochondria, channels which are called as the porin channels or porins. These are basically voltage gated protein channels. So these porins allow or help in the movement of small molecules, and here they do allow the fatty acids to move through. But the problem comes because inner membrane lacks these porins. So even if they get inside, they are going to get stuck in the intermembrane space and they cannot move further ahead. So this entire problem is easily fixed with the help of a Carnitine shuttle. Carnitine shuttle is a mechanism which helps us to move long chain fatty acids from the cytosol of a cell into the mitochondrial matrix. Actually speaking, for short chain and medium chain fatty acids, you don't need carnitine. Their movement is carnitine independent. But long chain fatty acids require a carnitine shuttle to help them move easily through the outer membrane and the inner membrane. Now this carnitine shuttle is comprised of different enzymes. The first enzyme which comes into play is the carnitine acyl transferase 1, which is also abbreviated as CAT1. This CAT1 is present on the outer membrane of mitochondria. There are also other names for this. For example, based on the fatty acid that it is catalyzing, there can be carnitine palmitoyl transferase 1 or carnitine acetyl transferase 1, carnitine octanoyl transferase 1. In general, we can call it as carnitine acyl transferase 1, or if you want to write it specifically, you can even write it as carnitine palmitoyl transferase 1. That is also correct. Now this CAT1 or CPT1, this is going to catalyze a reaction with fatty acyl-CoA, which was formed, and carnitine that is present inside the mitochondria. So I have carnitine being taken from inside the mitochondria combined with fatty acyl-CoA by this enzyme. So I have the carnitine and the fatty acyl-CoA combining with each other, and giving me two products. The first product is that I get back my CoA. So whatever CoA was formed or whatever CoA was taken during the activation, I get it back in the cytosol. That is what is shown over here. So that can be, the CoA can be written as HS-CoA, or you can even write it as just CoA. Alright, this is the CoA that I get back. And the fatty acyl part of it is now combining with carnitine to form a compound called as acyl-carnitine. So what I get over here is acyl-carnitine. Now, this acyl-carnitine can be transported across the inner membrane through or with the help of a transporter, what is shown here as carnitine acyl carnitine translocase. So in the first step, carnitine acyl transferase 1 enzyme combined the fatty acyl CoA with carnitine and gave me acyl carnitine. This acyl carnitine is transported, or it is moved across the inner membrane with the help of a transporter that is called as the CACT, carnitine acyl carnitine translocase. Now you might be wondering why I have given, or why there are, why carnitine is written twice. You'll come to know in a bit. So acyl carnitine now moves into the mitochondrial matrix, and that's what our main aim was, right? We wanted to get the fatty acyl CoA, or we wanted to get the fatty acid inside. So now the acyl carnitine has easily come inside the mitochondrial matrix. This part of it is done. But then the enzymes of beta oxidation will not act on carnitine. Carnitine was taken from the cell on loan. It has to be given back to the cell. So we have a third enzyme that comes into play over here. That is the carnitine acyl transferase 2, or CAT2. In case you have mentioned carnitine palmitoyl transferase over here, you can write the same thing over here. That is carnitine palmitoyl transferase 2. Or in general, like I said, instead of mentioning palmitoyl or octanoyl, you can just write it as acyl transferase. Now, CAT2 is an enzyme that is present on the inner membrane of mitochondria. This is very important to know, or to take note of where these enzymes are located. Now, carnitine acyl transferase 2, what it does is, it acts upon this acyl carnitine, what has been, transported from the outside. It acts upon that acyl carnitine, or it combines, or it works on the acyl carnitine, releases back the carnitine and adds a CoA group over there. So we have this acyl carnitine being acted upon by this enzyme. There is a CoA group which is taken from the matrix of the mitochondria, and now I get two products over here. The first product that I get over here is Acyl-CoA, because now the carnitine has been released. So I get Acyl-CoA, the same compound that I started with outside the mitochondria, and I get back the carnitine, because like I told you, the carnitine was taken on loan from the cell. I need to give it back to the cell. Now the carnitine which has been released is transported through the same transporter. You have to remember that it is being transported through the same transporter for the next cycle. Please remember this in mind, that this transporter transports carnitine as well as acyl carnitine simultaneously. So it is an antipode system. Any kind of antipode system is one wherein the molecules are traveling, or they are being transported in the opposite sides. Here you have the acyl carnitine coming in, at the same time a molecule of carnitine is going out. So this type of a system is called as an antipode system, where two molecules are moving in the opposite directions, and the carnitine acyl carnitine translocase is one such type of carnitine, or one such type of antipode system, wherein it is sending the carnitine from the mitochondrial matrix into the intermembrane space. At the same time, it is sending the acyl carnitine back into the mitochondrial matrix. So this is a very, very important transporter. In fact, the deficiency of the CACT, carnitine acyl carnitine translocase, has known to cause a lot of hereditary disorders which impair the fatty acid oxidation. So the people who have this hereditary disorder, they are unable to break down long chain fatty acids in their body, and usually they, such people will die within the first year of their life itself. So they are unable to break down any long chain fatty acids in the cell, which is a very serious thing. So that happens because of the deficiency, or it happens due to a problem, genetic problem in this translocase system, which is very important, because this is what helps in getting the fatty acid inside the cell. So now I have the Acyl-CoA ready with me. Now this Acyl-CoA is going to get into the steps of beta oxidation. So we have seen how the fatty acid moved from the adipose tissue, reached the target cell. Inside the target cell, it reached the mitochondria, and how it was able to get across the mitochondrial membranes into the mitochondrial matrix. Now this is where we are going to have the steps of beta oxidation occurring. We'll end the video over here. You can watch the video on beta oxidation at this link. Beta oxidation is the process where we have the actual breakdown or the catabolism of the fatty acid to give us acetyl-CoA. I hope you'll all join me in the other video as well. Thank you.